Good morning, everybody, and thank you for coming. We had a wonderful keynote talk from Jill to start the program today. And one of the things she mentioned was the importance of genetics and, how helpful it can be for patients, even without a, formal scientific background, to know a little bit about genetics. So that's where I come in. I'm Brad Williams. I work with the Jane Foundation, and I have a scientific background, although not in biology. So I spend my time at the Jane Foundation. I'm involved both in working on some of the scientific projects that we fund, as well as in patient diagnosis. Okay, so an outline of my talk. First of all, motivation. Well, Jill has already given you a lot of the motivation, but about why it's important to know something about genetics. And then I'm going to give some background about what genes are and what they do. And then what mutations are and how changes in your genetics can have an impact on function, such as causing dysferlinopathy or Emery-Dreifuss, as Jill talked about. And then I'm going to tell you how you can understand your own genetics report. If you look at the genetics report, you can look at it and, okay, this is exactly what that means. Okay, so why is DNA important? Well, for muscular dystrophies, such as dysferlinopathy, they're genetic diseases, which means that you can't really know someone has the disease unless you find mutations in the gene that's responsible for causing that disease. So everyone who has dysferlinopathy should have mutations in the dysferlin gene and not be able to properly make the dysferlin protein. Also, a number of treatments are being worked on, and you need to know, say, for gene therapy, you're going to give someone gene therapy. Well, you need to know what gene they need therapy on, first of all. But also, there's some other types of treatments, and one is a drug called adalurin that I'll go into more detail about later in the talk. And that will only work, and several other treatments being worked on will only work for someone who has a certain type of mutation. So some patients might benefit from this drug, other ones won't. So we need to know enough about everybody's genetics to know, okay, who can this drug work for? Who can be in a clinical trial to test if this drug really works? And then who should receive it, once it's developed and on the market. Okay, so a little bit of background on the dysferlin gene. Okay, dysferlinopathy is caused by mutations in the dysferlin gene which cause someone to not be able to properly make the dysferlin protein. Okay, dysferlin is a gene that everyone, regardless of gender, has 2 copies of. You only need one good copy to not have the disease. So, if, say, you are a patient and your parents and your children, unless, they inherited a second mutation somewhere, aren't going to have the disease. Okay, so we're going to talk about your mutations, but first of all, let's talk about my mutations. These are exactly what my mutations are. Reading this might not mean very much to you right at the moment. Wait till the end of the talk. Hopefully it will mean a lot more to you in about a half hour. And you're going to also know, okay, which of the potential treatments being developed might apply to me, and more importantly, from your perspective, which of the potential treatments being worked on might apply to you. Okay, so to begin with the basics, what are genes? Okay, think of the human genome as a giant cookbook, and each gene is a recipe to make a particular protein. So the Dysferlin gene is a recipe that tells the body how to make the Dysferlin protein. Okay, so what is a protein? Well, a protein is a big chain of these individual building blocks, sort of all stuck together like you would a toy, or one of my friends just pointed. Well, it's sort of like the barrel of monkeys game where you hook each monkey's arm onto the next one and pull them out of the big pile. Okay, so these individual building blocks are called amino acids, and to make a gene you need a long series of amino acids, and there's about 20 different amino acids that your body uses, and you need just the right amino acid in the right order to make the protein work as it's supposed to. And what guides the formation of each of these amino acids in the sequence, that's the information that the gene tells the body, this is how you make the Dysferlin protein. Okay, so one other thing is, if you can't make the Dysferlin protein, and if you have proteins in your diet, when you eat food that contains proteins, your body digests the food, it breaks the proteins into the individual amino acid, and then that's absorbed into your body. Then your body uses the instructions of the gene to assemble the protein. So even though in some sense the dysferlinopathy is a protein deficiency, it has nothing to do with how much protein is in your diet. Your body has plenty of ingredients for making dysferlin, but it doesn't have the recipe, so it doesn't know how to do that. Okay, so if you're a cell and you want to make a protein, here's how you make a protein if you're a cell. It comes in 3 steps. Okay, the first is called expression. Now, what that means is every cell in your body actually contains your entire genome, which means it contains the instructions for producing every protein that your body is ever going to produce. But not every cell produces every protein. That's what makes muscle cells different than skin cells, different than liver cells. So cells actually know, well, I'm a muscle cell, I need to make certain proteins to be muscle. So the expression is when the cell decides whether to make that protein. So if you're a muscle cell or a white blood cell, you know that you're supposed to make the dysferlin protein. Other types of cells in the body don't make it. Okay, now the way the gene is organized, it requires to be spliced together like a movie to make the final series of instructions to make the protein. Okay, so there's a bunch of splicing. I'll go into more detail in that because some kinds of mutations have to do with the splicing. Then after the gene is spliced together, it's translated, which means that the series of amino acids is actually assembled to make the protein. Okay, so this is an actual picture of the DNA and the protein amino acid sequence for dysferlin. The full thing is 2,000 amino acids long, and the DNA bases, which are, there's 4 different bases in DNA, A, C, G, and T, in, some very specific order. That's over 6,000 bases long. I'm not going to show you the whole thing. I'm just going to show you the beginning and the end. Otherwise, I'd either have to get a much bigger slide projector or make it really, really small. Okay, so the way this works is that each set of 3 letters, A, C's, G's, and T's, forms what's called a codon, which is kind of like a word of DNA. And each codon stands for a particular amino acid, one of the 20 amino acid that your body uses to assemble the protein. So as your body reads through the gene, this is after it's all been spliced together. It says, okay, the first codon is ATG. That stands for the amino acid abbreviated as M. I'm not going to say the full names. Then the next one is CTG. That stands for the amino acid abbreviated by L. And it keeps going on for 2,080 amino acids. Then it gets to this codon at the very end, TGA. Okay, that's a different kind of codon. That's called a stop codon. And what that says, it doesn't tell the body to add another amino acid. It says, okay, this is the end of the protein. We're done here. You're finished the protein, and it's ready to go. And that's actually important because there's a type of mutation that I'm going to come back to and talk about that a little bit more. Okay, so if you have changes in your DNA from the standard sequence, there's a sort of a vocabulary that's helpful to know in understanding genetic reports. Okay, first is the word variant. Variant just means any difference between your DNA and sort of the average, DNA for a typical person. It turns out none of us are average. Every one of us has various variants in our DNA. That's what makes us all unique and different from each other. Okay, variants are not always harmful. They can be things like having straight hair or curly hair or brown or blue eyes. None of those things are harmful. In a case where it's just makes you different but doesn't cause any harm to your body, it's called a polymorphism. Now, if it might cause harm to your body, it's called a mutation. So that means that it's a change in DNA which makes your body unable to either make some protein or it makes the protein in such a way that it isn't going to work as it should. And the other thing that you might see if you look at a genetics report is the word VU, variant of uncertain significance. That's a fancy way of saying there's a change in the DNA. We're not sure whether it's a polymorphism, which would be harmless, or a mutation, which means it could potentially cause disease. And we'll talk about situations that we tend to not be sure about and say that they're VUs on the genetic report. Okay, so we're going to now go over the different types of mutations that are commonly seen in DNA and in people's genetics. And because reading thousands of A's, C's, G's, and T's isn't very intuitive for humans, I'm going to use an analogy. And that is going to be, we're going to have a sentence made up of 3-letter words. Why 3 letters? Because every 3 DNA letters is a codon, which is kind of a word of DNA, which stands for a particular amino acid to put in the protein. Okay, so our sentence that's going to be our model for illustrating mutations is going to be, the cat saw the rat, but not the bat. And then it ends with the word out. Out is going to be our version of a stop codon. Think of it as being like saying, ending a conversation by saying peace out or over and out. That's not actually part of the sentence, but that's just our code word for saying, okay, this is the end of the sentence. Now, the other thing we need to realize about DNA is, unlike when we write words, we put in a space to make it easier to tell where one word ends and and the next word begins. DNA isn't like that. Words are always 3 letters, so when your body is reading the DNA, it just says, okay, take the next 3 letters, that's the next word. So if there's not a definite spacer to say, okay, this letter is always the beginning of the word, and that's going to be important. We'll elaborate on that a little bit later in the talk. Okay, so the first thing I'm going to show is an example of a polymorphism. So remember, polymorphisms just make you different. They don't cause you any harm. So if you spelled the word cat with a K, it's different. If you do it in school, your teacher might take out the red pen and make a mark on your paper. But, we all can read the sentence and we still know what it means. It's still intelligible. So, okay, now we're going to sort of illustrate what happens with the actual dysferlin protein. We're going to draw the normal dysferlin protein on the bottom as a happy little cloud, if you've watched any of these TV shows about painting. And then our version with a slightly different spelling is still the same shape of happy little cloud, but it's got a star on it indicating that it's different. Okay, now we come to our first class of mutations. Now, this is called a nonsense mutation. Now, we think that you probably all know what the word nonsense means in ordinary conversation. It's something that's just gibberish. It doesn't mean anything at all. Well, in genetics, nonsense actually has a somewhat different meaning, and it has a very precise meaning. What it means is that you have your change in your DNA causes there to be the stop codon, which marks the end of a gene, in a place where it's not supposed to be. Okay, so for our example sentence, we're going to change in the word but in the middle of the sentence, we're going to change the B to an O. Okay, that changes the word but to out. So what happens when the body is reading through the sentence? It says, the cat saw the rat out. Okay, out. That's the end of the sentence. So it thinks that the sentence is just the cat saw the rat. It doesn't see the last half of the sentence, but not the bat. Okay, so we're going to have only make the first part of the gene of the protein, and that might be completely useless to the body. So now we're going to, instead of our happy little cloud, we're going to show kind of a scrunched up little cloud, and the body might say, that's no good, I can't do anything with that, and just sort of throws away the fragment of the protein because it doesn't know what to do with it. So that's our sentence example of a nonsense mutation. Now, what does a real nonsense mutation in dysferlin look like in a genetic report? Okay, this is an actual mutation. It's actually had a paper written about a number of patients who have this mutation. Okay, so what does this notation mean? I'll start talking about that. Okay, the letter C at the beginning stands for cDNA. That's basically DNA after you've done all the splicing together to get the protein sequence. So that's what's happening with the actual DNA. And so in this mutation, you change the DNA base number 6086 in the gene from a C to a T. Okay. Okay, now, and then the second half of this stands for P for protein, and this shows what the consequence of that genetic change is going to be in the protein sequence. Okay, so we make this change in a single base in the DNA. Okay, what does that do? Well, that codon that DNA base is part of is amino acid number 1905 in the dysferlin protein. Okay, normally that's the amino acid arginine, which is, we'll just call it R. And what that does is it changes that codon. Instead of saying, I need an arginine here, it says, this is a stop. This is the end of the protein. Okay, stop is, I'm not going to make you remember the letter codes for every amino acid, but one you should remember is a stop, and that's an X. Okay, so instead of, when it gets to position 1905, that's about 150 amino acids before the end of the protein, it says, okay, we're done here. It doesn't make the last part of the protein, and it's not useful at all to the body. But the drug adalurin, which I mentioned before, it's been in clinical trials in Duchenne muscular dystrophy and cystic fibrosis. What it does is it tells the body to run stop signs. Okay, so if you have a stop mutation, it makes the body sort of, this isn't really, maybe I'll just keep on going and keep seeing if there's more amino acids to throw in. Well, if you have a stop where you're not supposed to have a stop, then the body's running the stop sign is actually a good thing. That might enable your body to make the full version of the protein, and that would be useful for you. So if you have a nonsense mutation, then this is still being tested. We don't know 100% how well it's going to work, but if it does work, or something like this works, then that's a treatment for you. So what you want to look for to see if you have a stop mutation, there's going to be a letter, in this case an R, any letter but X, then a number, and then an X. That's in the protein part of your mutation. Okay, the next type of mutation we're going to talk about is called a missense. Now, this is also just changing one letter of the DNA, but here it does a different thing. We're going to change the word in our sentence example from an R to an H. So that makes the word rat be hat. Okay. It doesn't stop the sentence midway through, but it does change the meaning of it. So our cloud is going to be a little bit weirdly shaped. The body may or may not like it. Okay, now a little bit of an aside on missense mutations. Okay, there's over 2,000 amino acids in the dysferlin gene. Any one of those amino acids can be switched out for 19 other amino acids. So if you think of all the switches in amino acids that's possible, and it's a very large number. We don't always know what switching any one of those amino acids to another one, it may be a polymorphism, harmless, just makes you different. It doesn't cause disease, or it may cause disease. So one situation in which a genetic report might have a VU, variant of uncertain significance, is if it changes an amino acid, but we're not really sure whether it makes, what effect it has on whether the protein does what it's supposed to. As we test more people and gain better knowledge of genetics, we'll understand more, but it takes a while. Okay, so in the genetics report, this is actually pretty similar to the last one, in that we also, in a certain place in the gene, again, we change a C to a T, but here we started again with an R, but now instead of changing it to an X, which would be a nonsense mutation, we change it to a different amino acid, which is abbreviated by a W. So we just switch amino acids. It turns out this particular change in amino acid is a mutation. It does cause disease. It's been found in patients. Another type of thing that you can do with the DNA is you can add one or more letters, just stick extra letters into the DNA. Now, if we stick in 3 letters, then remember each 3 letters is a word, so actually we stick another word in, only here we're sort of just sticking 3 letters in the middle of the word, but beginning with the word cat, the grouping of letters is still the same. But you're going to have a couple sort of nonsensical sounding words at the beginning of the sentence. When, in the analogy in an actual protein, the protein is going to have some weird stuff going on. Our cloud is going to be a little bit funnily shaped, and your body also may not like that. Okay, so how do we write an insertion of bases? So in the DNA, we have the word INS standing for insertion, and here it's between base 3505 and 3506, and we stick these 3 bases CGC. And what that does is that adds an extra amino acid into the protein. You don't need to remember all the details of this, but the thing is, if you stick in three bases, you add an amino acid. Now, what happens if you stick in 1 or 2? Okay, so this is an example. We're just going to stick in 1 base. You might think, well, just adding 1 base is going to be, less significant of a change than adding 3. Well, it turns out that's actually not true. Because there are no spaces in DNA, everything is grouped in 3 letters at a time. So if we just add another thing, or dividing it into 3-letter words, in our sentence example, we get things that aren't really even words. Now, in DNA, every possible combination of 3 letters is a word. It specifies an amino acid, but every amino acid beyond that point in the protein is going to be the wrong one. Which means depending on, you might have hundreds or thousands of wrong amino acids. Well, that's not going to be much good. So you're going to end up with some really weird looking cloud here. The body is not going to like that at all. Okay, so this is an example of an insertion mutation. It has the INS here in the DNA part. But what it has in the protein part, it has the lowercase fs. That stands for frame shift. That means that you've, added something that isn't a multiple of 3. So beginning with amino acid 1169, it's changed from an L to a Q. But really what this means is that amino acid 1169 and every amino acid after that is going to be the wrong one. The other thing is usually when you have a frame shift, at some point later in the protein, you'll have a stop codon. Just because if you have sort of random sequences of DNA that aren't really encoding for a real gene, you tend to have a lot of stop codons. But this isn't a nonsense mutation. The stop codon only appears because you're grouping the letters together wrong. Okay, now the opposite a special case of an insertion would just be a duplication. And the only difference between an insertion and a duplication is just that you take, some number of letters of DNA and then just repeat those one or more letters an extra time. But really, it's pretty much the same thing. Again, it makes our cloud a little bit weird if it's not frame shifting, but a lot of times it is frame shifting. This example, which again is a real mutation that's been found in patients in Dysferlin, there's an A at position number 1392 in the DNA, and you just repeat it an extra time, so that causes a frame shift, which means that everything beyond that point of the protein is going to be completely wrong, and you're going to eventually run into a stop codon, and that's not going to work for your body. Again, and this is sort of just the opposite of an insertion, you can take out DNA bases. Here in our sentence example, we'll just take out an entire word, the word rat, and now this sentence isn't even grammatically correct. The cat saw the what? And so the cloud is missing something and is the wrong shape. Your body, depending on how important that word is, your body might, be able to sort of do something with it or not at all. And most of the deletions that we are found in patients are things that are maybe one or two bases, and they tend to also be frame shifting. So any number of adding or taking away bases that isn't a multiple of 3 is a frame shift. So this example is yet another frame shift. The word deletion is abbreviated by DEL in the DNA, and the effect on the protein is pretty similar to what we've seen in the last couple of examples. Okay, so now I'm going to talk about one other type of mutation, and this requires stepping back a little bit and giving a little bit of a background. This has to do with the way that the genes are spliced together. So sort of an analogy of this is that assembling a protein is kind of like making a movie. When people are making a movie, they film a number of different scenes and different camera shots, and then when they've filmed all this stuff, then they splice things together in just the right way, to tell a story that you can watch and show in the theater and hopefully find a big audience. The other thing that actually happens in proteins is that you can make more than one version of a movie. Say you saw the Lord of the Rings movies in a theater, but you're a big fan, you buy the DVD, and then you'll find, I get a bunch of extra scenes that, if you're a true fan, you'll really like this, but if we'd put all of those in the theater version, that would have made the movie way too long. The theater wouldn't have liked that because they couldn't have shown it as often. So there's different versions that can be made of the same movie. In the same way, the body can actually make different versions of proteins by splicing the DNA together in different ways. There's actually a number of different versions of, slightly different versions of dysferlin. Okay, so I'm going to introduce a couple new words. The parts of the protein, the parts of the gene that are included after splicing that actually tell the gene the body how to make the final version to make the protein are called exons. So these are regions that have these 3-letter codes specifying an amino acid that I've just shown you. The rest of the gene, the parts that get spliced out, which are thought to contain directions on the body on how to make the gene, how to do the splicing, when to splice it together in different ways, are called introns. One little fact about dysferlin, 97% of the dysferlin gene, as it's in your DNA, is actually introns. So only after you splice it out, it's only 3% as big as it was initially. So there's a lot more introns than exons, and they have to be spliced out correctly. So the locations where, so one kind of mutation has to do with affecting the splicing. Okay, in our sentence example, I'll give you a slightly different version of the sentence. So this is the sentence as it would exist in DNA before splicing. So we have the first two words, the cat, of the sentence, and then we have a bunch of letters that aren't part of the sentence. And then we continue with the sentence, saw the rat, etc. So these letters that aren't part of the sentence are the intron, and then the words that are part of the sentence are two different exons. These letters in yellow that say, splice begin and splice end are actually directions on how your body is supposed to do the splicing to put the sentence together correctly. And this is actually what happens in DNA. Near the beginning and the end of each intron, there's DNA bases that don't say what the protein is going to look like, but tell the body how it's supposed to do the splicing. The way it's supposed to happen is that the body sees, okay, these things printed in yellow, okay, splice begin, splice end, okay, I take out that whole part, put the exons together, and okay, now it's the sentence that we saw before. The cat saw the, etc. Okay, but what happens if there's a mutation in this yellow region? We change the L in splice to a V. Now it's not splice. Your body's reading it. It doesn't see a word splice. It says that's not a splice. I guess that's just part of the sentence. So now it's going to group all of those letters into 3-letter things. There's actually a few words in there, but really it's gibberish. It doesn't mean anything, and your body isn't going to be able to do anything at all with if it doesn't splice things correctly. Okay, so the reason for elaborating a lot on splice is splice mutations are actually very common. They're a very common kind of DNA mutation. Okay, so here I'm going to give you a couple of examples, and I'm just going to show you the DNA part, because it turns out if you have a splice mutation, you never really get as far as even making the protein. Okay, so in this one, remember the splice is actually in the intron. It's not in the part that actually codes for the amino acids in the protein. So the way the numbering goes is you take the nearest base that's actually part of that encodes for the protein, and then you count into the intron. So this would be 855, which is a base which is in the splice DNA, and then you count one base into the intron. And in this mutation, that gets deleted. The body doesn't recognize that it's supposed to splice there. You can't make protein. This is another one. Now this is, here the nearest base that's part of an exon is 3349, and here and that's right at the after an intron, and here you're counting backward 2 spaces into the intron, and you change the A to a G. This also affects splicing. There's a lot of details, but what is helpful to remember in a splice site mutation, if there's 2 numbers separated by a plus or minus, that's your signal that the mutation is in an intron, not an exon, which almost always means that it's a splice mutation. Now another reason that we have variant of uncertain significance, in genetic reports, we don't know if something is a mutation or not, has to do with splicing. We don't completely understand splicing, and it's a little bit hard to predict if you have some DNA base in an intron which changes. Sometimes you know good and well that it's going to affect splicing. Other times you're not so clear, and you have to just see what happens, what the body does with that variant, if it's still able to splice correctly. So one common cause of having a VU is that it might affect splicing, but we're not sure. Okay, so that's the end of our examples of different mutation types. Now I'm going to go back to my mutations, and now that you've seen some examples, we'll actually talk about what all of this means. Okay, so the first one, what kind of mutation is this? Anybody have a guess? Duplication. Okay. The key is, well, I said it in the comment, but it's a DUP on the thing. Okay, and there are actually 5 bases, 879 through 883, which are duplicated. Okay, what is the effect of that going to be? Well, is 5 a multiple of 3? No. Yeah, okay, so that causes a frame shift. So from amino acid 295, which is actually quite early in the protein on, it's going to get everything wrong. And in fact, after about 45 codons, it runs into a stop because it's reading things in the wrong order. Okay, so I'm not going to make protein like this. Okay, now my next mutation, this is on the other copy. Copies are called alleles in genetic speak, but we can just call it a copy. So this is, the first one actually was the one I got from my mother. This one is the one I got from my father. Okay, so this is a deletion. This is a really big deletion. This is a deletion of 179 bases. And it turns out this is, I'm just completely missing an entire exon. It actually took me a few years to find out about this mutation after the other one was discovered. So the way they actually sequence DNA in a genetics test is because most of the gene is introns, you just want to save yourself work and just sequence the exons. And you look for any sort of spelling errors in the DNA in the exons. But if you're just completely don't even have an exon there, nothing is going to show up from that copy of the gene. It's just going to see the other copy, which is fine in that exon. So you can't actually do that. So they actually had to do a different kind of genetics test, to see whether I had, just one copy of that exon versus two, and they said, yeah, you only have one copy of that. And then they actually analyzed my father's DNA and said, yeah, you've only got one copy of that too, so that all makes sense. Okay, so again, now just missing an exon isn't always a terrible thing. There's actually some drugs which are being tested for DMD, which cause people, if they have some mutation in a given exon, you might be able to just throw out that exon, just skip over it entirely, and still put the rest of the protein together and have it, be somewhat useful for the body. Okay, in the dystrophin protein, which is for DMD, that actually tends to, there's an exon which a lot of people have mutations in, but if you just skip it over, you have kind of a mild form of Becker muscular dystrophy, much better than having Duchenne. Okay, but in my case, I did not get so lucky. 179 is also not a multiple of 3. So once again, I get a frame shift. So every amino acid after that point in the protein is wrong, and I run into a stop. So, okay, so that exon isn't one you can skip because skipping it would cause a frame shift. Okay, now the other thing is because I have one mutation in one copy of the DNA and a different mutation in the other copy of the dysferlin gene, both of these are heterozygous. That means you only have one copy of that variant. Now, someone who has 2 copies of the same mutation on each of their dysferlin gene would be homozygous. So just another word that gets thrown around that it's useful for you to see. Now you have a glossary handout in your packet, so you don't have to remember all of these words from right now, but you will be able to watch the video later. Okay, so to review, this is essentially all of the things I'm going to talk about. The words that refer to changes in DNA, okay, a variant. That just means any change regardless of whether it's bad or not. If it's harmless, it's a polymorphism. Incidentally, when Jill in the last talk talked about how different people with the same genetic change can have very different presentations or different symptoms. A lot of that is thought to be due to other polymorphisms that individuals just happen to have, which can modify the course of the disease. And that's kind of a very active research area. Okay, if something does cause disease, that's a mutation. A VU is when we aren't sure whether it causes disease, but hopefully we will be in the future. Okay, now a review of some of the mutation types. Okay, so I'm going to break this out into mutation types in the DNA itself and in the protein. Okay, so in the DNA you can have one letter change to a different letter. That's a substitution. You can add, first you can delete one or more letters. That's a deletion. You can add one or more letters. which is an insertion, or as a special case of an insertion, you can repeat one or more letters. That's a duplication. So those are kind of the four common things that are worth remembering. Okay, at the protein, what the effect of the DNA change can be, it can affect splicing, so that's a splicing mutation. And remember, in the splicing mutation, you have a plus or minus usually in the numbering. You can change one amino acid to a different amino acid. That's a missense mutation. You can change an amino acid to a stop. That's a nonsense mutation. Or you can disrupt the grouping of 3 DNA bases in each word. That's a frame shift. There's going to be a quiz. Hopefully you've remembered all of that. But this isn't going to be a high pressure quiz. You're not going to get a grade, but you might get a prize. Okay, so we have 2 questions on this quiz. Here's the first one. And this is an actual mutation from a patient in Dysferlin. And so there's 3 things I would like, any of you who volunteer. What kind of mutation is this at the DNA level? The first part of this. What kind of mutation is this at the protein level? The second part in parentheses. And would adelurin, that's the drug that I talked about, potentially be able to treat this mutation? So I will take volunteers. Okay.
It's a nonsense mutation?
Okay, which part is that in the A or B? At the DNA or protein?
Oh, at the protein.
Okay, and then what is it at the DNA?
Maybe I don't understand the question.
Okay, so what type of change is it to the DNA?
Oh, it's going from C to T? Is that what you're asking?
Yeah, but
okay.
But what kind of...
Oh, a substitution?
Substitution, okay. And would adelurin potentially be able to treat this mutation?
I believe so, yes.
Okay, yeah. Excellent. Very good. Okay, congratulations. Okay, and we have a second question. Okay, it's essentially the same things that we're asking, but about a different mutation. Who would like to try this one?
So this is a duplication?
Yes.
And in the DNA level, it's a duplication. In the protein level, it will be a frame shift to create stop codon.
Okay, yeah, it's a frame shift. So could adelurin treat this?
Yes.
No. Well, okay, we're going to have another volunteer. If anyone can explain about whether adelurin can treat this. Okay, we have one over here? Okay.
It cannot because it's missense and not nonsense, and nonsense only works, that drug only works for nonsense mutations.
Okay, you're half right. It's it does only work for nonsense, but is this actually a missense? Okay, okay, let Sarah come up.
So the reason I believe adelurin probably would not work is because it would not correct the frame shift, even if it's corrected the stop. So you would still be frame shifted throughout the rest of the protein.
Yes. Okay. Okay. So thank you everyone for playing this quiz game.